Hello, and welcome to our final lecture on microbial nutrition, where we get into the fun kind of naming kind of concepts that we have. Um, and so here we're going to see the effects of pH on microbes. Okay, and it's going to kind of be simple to think of like maybe there's a few different types of these um, bacteria. And it will make sense, right? Like if there is an acid and there is a base in life, then things probably like bases as their environment and things like acids in their environment. Okay, and so that holds up in the case of lactic acid bacteria. Um, uh, there are species of bacteria called lactobacillus that we use when we ferment milk into yogurt and other types of lactic acid bacteria to make sauerkraut, salsas, gazpachos, um, and all kinds of unique tangy dishes that we like. Okay, that tanginess um, is probably a useful mechanism to our brains, right? To where saying, hey, if this is tangy, it kind of has a pleasant taste to us that we keep eating it because it's telling our brains that it likely doesn't have bacteria in it, like the bacteria that can make us sick. Because if we're eating something acidic or tangy, then it is not a mesophile or something that's just in the water, right? And that mesophile is the thing that is going to infect us. Let's get into it. Okay, so I say mesophile, meaning the general term of uh, it likes to be happy in the medium, right? It's in the middle. So mesophile can be with temperature, can be with other things, but you can have more specific terms like neutrophile in the terms of pH. So we have acidophile, alkalophile, and neutrophile. Okay, acidophile being the easiest one there. An acidophile means someone who loves acid. So a bacteria who loves acid would be our lactobacillus friend that we just talked about. The ones who like the neutral pH, or the ones who can infect us the highest, right? Look at this. E. coli, Staphylococcus, and Salmonella are in that neutral range, right? Like that just water pH range. We forget water from the lakes, the streams. Um, we have a high chance of being infected with that water, unfortunately. As great as it would be to just drink natural water. Then alkalophile, if you've heard of alkaline batteries or alkaline solution or Alka-Seltzer, um, just means that it is a basic solution. Why does it work against heartburn? Heartburn is a lot of acid. Alka-Seltzer brings in a base, and it's going to neutralize that high acid content. Uh, bring it up more neutral, even if it is still acidic. All right, so alkaline, alkophile. Okay, so that is how we grow in pH. And so before I move on, let's explain this. What's happening in this graph? Okay, if you can hear me say acidophile, neutrophile, alkalophile, but what am I actually talking about? So if we look at our y-axis, that's growth rate. How fast do things grow? Basically, is our generation time 20 minutes? Is our generation time 30 hours, right? This is what that's saying. How fast do they grow? But we're going to keep everything the same. We're going to keep temperature the same. We're going to keep agitation the same. We're going to keep everything the same. Only thing that we're going to change is uh, the pH, okay? And so we're going to stick a piece of lactobacillus culture in pH 1 and watch that nothing grows, okay? Not a thing, okay? I don't even need to try a neutrophile, okay, because even at 6, they don't grow. I don't even try an alkalophile because even at 7.5, they don't grow, right? So you see what's happening now. Okay, so then I raise up my broth to 2, and that's how much they're growing. They're going to be that much overnight. Raise it up to like 3.2, 3.5, and I have a lot of growth, okay, when I add lactobacillus, okay? Now, the thought experiment is if I still added E. coli at this pH, because at this pH it doesn't have any growth, it still wouldn't have any growth there, right? Okay, so bear that in mind. That's the thought experiment behind it. So this is why you have this blue graph, because those are the 3 species that represent that blue graph, 3 species that represent the green graph, and then finally these are the species that represent the alkalophiles. So at pH of 11, even they stop growing, okay? And they grow best at pH 9. All right, so look at the top of your curve, look at the apex. That's the best growth that they could possibly do. Okay, look down. What's your pH? Are you in the acid range, 1 to 7? You are probably a acidophile. If you look down and you see you're at 9 to 12, you're going to be an alkalophile. Okay, and then obviously you can make the judgment calls around neurophiles. Alkalophiles kind of go with halophiles here. Okay, we're going to go with halophiles in just a second. But that's what you see here, a alkalophile and a halophilic bacteria causing that color. Okay, temperature can also affect things. Okay, and these organisms, these tube worms, grow, and also microbes grow. Down there, archaea, like we said. All right, and so here's some terms you're going to want to know. All right, so psychrophiles, okay, note the temperature. We're going down here at 10 degrees Celsius. That is almost freezing, okay, so you can put those bacteria in the freezer and they would, well, in the fridge, and they would still be alive, okay? So those are things like listeria, okay, that we just talked about. Listeria would be a psychrophile because it lives in the fridge just fine. Okay. Okay. Mesophiles, we talked about temperature and how they like temperature. Okay. Most things that affect us are mesophiles. Okay. It can be temperature. I like to use it for all of them because it's just kind of middle of the road bacteria are the ones who are going to get us. Thermophiles. Okay. If we look down at the top of our graph, 65 degrees Celsius. Body temperature is 37, which is where our mesophiles love. So 62, 67 degrees is super hot. Okay, those are the ones that live down near those springs. Okay, and then the ones that live at those vents in Yellowstone or deep under the ocean in Hawaii would be hyperthermophiles. Remember, hyper, if you give your kid too much sugar, they're going to be hyper, right? So if you give it too much temperature, you're going to be a hyperthermophile. Okay, then we're going to end with some other factors that affect growth. First, we have halophiles and barophiles. We've talked about halophiles pretty extensively. Okay, salt-loving creatures, right? In the Great Salt Lake, you get this purple bloom, okay, from an algae, okay, and the Archean halobacterium. It's even named after loving salt. Okay, halo means salt in Greek. Halophiles like salt. They're the Great Salt Lake. Picture that was in one of our other slides. Any kind of colored water that you see is due, well, in microbiology, is due to these halophiles or some other kind of cool growing species. Okay, and then we have barophiles. Remember barometer? Okay, if you ever heard of barometer, the low pressure system's moving in. Let's check the barometer. It's like a weather thing, right? We're checking pressure, the pressure of the atmosphere on us. Okay, so things that can live under high pressure, right? All the way at the bottom of the Titanic, right? That was in the news recently. But there are bacteria that can live down there, right, and that are decomposing that ship as we speak. Um, and those would be barophiles. Okay. We also have osmophiles and xerophiles. Okay, and we have some candy over here. Why are we dealing with candy when we're talking about osmophiles and xerophiles? Well, okay, osmophiles can live in places with high sugar. Okay, remember when we talked about the cell and osmotic pressure? What's going to happen if you have a whole bunch of sugar on the outside and very little water? Well, meanwhile, you're a cell with a little bit of sugar and some water in it, right? What will happen, right? Very little water on the outside, high solutes on the outside, right? So it's going to rush out. All that water inside your cell is going to rush out to try to balance out the water and solutes and sugars, okay? So that is bad, okay? So uh, typically osmophiles can withstand that osmotic pressure, right? Osmotic pressure, osmophile, kind of makes sense, okay? They can withstand that osmotic pressure and not lose their water, okay? So why am I including it with xerophiles who can take dry, desiccated environments? Well, dry, desiccated environments are like candy also. There's not a lot of moisture in the candy, okay? And what little moisture there is fighting against the osmotic pressure, okay? So that is why when you look at this question down here, candy can be stored at room temperature. Okay, it's also cost efficient, right? To leave it there and you're going to buy it when it's right in front of you rather than if it's in a fridge, right? We get it. All those reasons stack up too, but the reason why you never open up a package and it's disgusting is because the rarity of getting osmophiles and xerophiles to land in your bag of candy during production, because they do use very sterile conditions, sanitary conditions, beard nets, all the good stuff. So in order to get them in there and to survive is rare. Okay, we'll end with some of the bacteria media that we used, right? We talked about these terms: enriched media, selective and differential media in the lab. Be sure to look over these again. I'm going to hammer it home here. Selective media selects for a different type. Certain things won't grow on it. Differential will make things look different. Okay? All right. Enriched media makes things grow that are called fastidious. What's fastidious means? It means stuff that is hard to grow. Okay? And remember, one of our very, very important selective plates is the McConkey agar plate, which differentiates between fermenters and non-fermenters of lactose. Okay, you wouldn't waste your time putting something that's not gram negative on here, but once you have something that's gram negative, you would put you would look and see if the pink color is there, and that means they ferment lactose, or you would look at this yellow color here, and that would say that they do not ferment lactose. Okay, so how they ferment sugars is another type of classification scheme, right? So remember, all of these things are ways to classify bacteria, okay, and ways to organize bacteria. You want to remember how they grow and how they live. And that is the end of our nutrition lectures.